Thank you very much for the invitation. I'm really sorry I cannot be there in person. Of course, I'm going to say a sentence now with absolutely zero information content, that the immune system is also a system to which Giorgio has made contributions as to essentially anything else we can think of. So let me try and tell you about the immune system in a somewhat painless way, assuming you're not all immunologists. So imagine you're at a meeting, just like the one you're at right now, in a remote location, so probably more remote than you're finding yourself right now. Everybody's excited. In the evening, there's a party, and the next day, Joe is found dead. And next to Joe's body, there's a few drops of blood that don't belong to Joe. So these being scientists, they shout genetic tests for everyone, which they quickly carry out. And this identifies that the murderer is one of two twins, either Alex or Terry. So, of course, genetic tests are DNA, and identical twins have exactly the same DNA. So they're unable to a first approximation, distinguish between identical twins. So what can we do? So they call in Detective Aziabe, who suggests to consider a new modern forensic technique based on the immune system. So what is the immune system and an adaptive immune system? You all have an adaptive immune system. It's made of B and T cells, otherwise known as lymphocytes. Probably after the last few years, you're all very well aware that you have lymphocytes inside you. And the role of these cells is to recognize foreign pathogens. They do it using these receptors that they have on the surface. And these receptors basically bind different molecules in the body, and they're able to figure out which of these molecules is natural to your body, so you should leave it, it's okay for it to be there, and which one is foreign, meaning it's probably attacking you, it's a pathogen, you should get rid of it. So they're able to perform a discrimination task. However, although one cell has many receptors, they're all of one type. So to protect you against the many different pathogens that are out there, you need a diverse ensemble of different receptors. That means different cells. That's what we call an immune repertoire. And when a cell or receptor recognizes a pathogen, it proliferates. It makes many copies of itself, and then you have a bigger army of the cell that is able to recognize the pathogen to, well, basically quench the infection. And after some time, cells die. So you see that we have the statistical system where basically thanks to the statistical properties and the diversity of this system, it's able to protect you from pathogens. And recently, thanks to sequencing technologies, we can go in, get a blood test, sort out the B or T cells, so the lymphocytes, sequence the receptors that are proteins, and learn about this diversity. So we have an insight into this diversity. And also, thanks to the fact that after an infection, these cells proliferate, then they go back down. But basically what we're carrying around in us is a personalized medical record about which, if we could read it, which we're trying to learn how to read it, we couldn't lie about our medical history. Everything's there. Everything that happened to us, both vaccines and real diseases, are in there. So that's the big name of the game. How do we learn to actually read it? Okay, but how is this going to help us solve the murder mystery? Well, we're going to take the blood sample found by Joe's body and compare it to blood samples from different people, for example, our two twins. And we're going to be able to figure out if the two blood samples we're comparing, the one from Joe and the other one, come from the same person or from the different one. And how are we going to do that? Well, we're going to do that by just looking at the number of shared sequences between the two samples. So again, we're using a statistical measure to figure it out, because it turns out that if the two samples come from the same person, they share many more immune receptors than two samples that come from two different people, even if these two people are identical twins, which we see here in pink. So there's an order of magnitude separation here. So basically, you share many more sequences with yourself, even in different samples, than between any two people. And this signature is stable over time. So this allows us to identify the murderer as Alex, and it'll allow us to identify the murderer even 40 years after the murder. Okay, so, but this being scientists, they quickly ask, well, how does this work? Can you explain the details? So the first thing to realize is that you have of the order of 10 to the 9 different T cells in your body. As I said, they come in clones, they come with populations, but if we only look at unique ones, you have over the order of 10 to the 9 different receptors. Now, as you know, probably proteins are encoded in genes in the DNA. So if we want to encode 10 to the 9 receptors, we need 10 to the 9 genes. But actually, we have of the order of 10 to the 4, 10 to the 5 different genes that we fit the DNA into this organelle called the nucleus. And so if we wanted to encode the receptors, we would have to fit 4 or 5 orders of magnitude more DNA, and that physically would not fit. That means this nucleus would have to be, instead of being the size of 1/10 of a pixel, it would have to be the size of 2 millimeters, which is a crayon. And that means that our body would have to be 20 meters high, which is the size of a giraffe. So that doesn't work. We have a problem of how to fit it, and that's obviously not what's going on. Instead, nature has come up with a funky way to generate these diverse receptors, which combines combinatorics and randomness to generate diversity. What does that mean? Well, each receptor, to a first approximation, is made out of 3 parts, which are called V, D, and J. And in our DNA, we have templates for these 3 parts, a certain number of templates for V, D, and J. And then in a combinatoric process, you edit the DNA, you cut it, and pick 1 V, 1 D, 1 J, which gets us, if you look at the combinatorics of this process, to the order of about 1,000 different combinations. But we need to get to 10 to the 9th. So we need a few orders of magnitude more. And that happens through random, non-templated insertions and deletions of nucleotides, or simply letters, in the DNA between these gene templates. So you put together 3 building blocks, and then you take out and put in different letters. And so just to give you an example of how this works, imagine you have 2 building blocks, which are called biological physics. What this process is that you can take out and put in any letters in between, basically at the junction between these 2 words, which makes biological physics into biotic aesthetics, or biopic logistics, or biology basics, and so on and so on. So you see how this process can easily generate diversity. And we're basically dealing with a molecular version of this word game. And you can go on and on and on and do this. So the question now is, can we characterize the statistics of this random process? And the first thing to realize, based on the example that I just showed you on the word example, is that if I give you these 2 final words, like biopic logistics or biology basics, you have a hard time figuring out what was the initial words that they came from. And the same is true here. If you want to figure out what was the initial way, the genes that they came from, it's essentially impossible. If you want to figure out what were the building bricks that went into it. However, it's impossible in a deterministic way, but we can do it in a probabilistic way. So that's what we do. We take each sequence and we build a library of all possible scenarios, by which I mean V gene choice, DJ choice, J gene choice, and the insertions and deletions at each of the junctions. We consider all possible ways of generating this sequence, so all possible scenarios that are consistent with the sequence we're considering, assign weights to them, and then sum over all the sequences to calculate marginal distributions, such as the probability of using a given gene, the number of insertions, deletions, and so on. And then we have a model, the most general factorializable model that we use to recalculate these weights, to calculate the probability of each of these scenarios. And we do this until convergence. It's proven that this procedure, which is based on expectation maximization, will converge. And at the end of the day, we get weights for each of the processes. And then we just have to sum over these weights to calculate the overall probability of generating a given sequence. So either you're familiar with these kinds of Bayesian approaches, or let me just give you the takeaway that we do it in a probabilistic way. But we're able to figure out, based on data, sequencing data, what is the probability of generating each of these sequences. And this is what it looks like. So if we now look at the histogram of the log probability of generating each of these sequences, so we ask how many sequences do we have that have a probability to be generated, that's 10 to the minus 10, oops. Sorry, 10 to the minus 10, 10 to the minus 12, and so on, we get a histogram like this. And the first thing to notice is that it spans over 20 orders of magnitude. So there are some sequences that are relatively easy to be generated, given that we generate more than 10 to the 9th, and some that are quite rare. The other thing to notice is that the red line here is the variance of the order of 1,000 different people, real people like us. And you see this process is incredibly reproducible. So in all of us, the probability of generating a given receptor is essentially the same. Okay, so now this gives us power. We've learned the stochastic rules for the process. So now this gives us the power to explore how the repertoire functions. And then we can look at the naive repertoire, because what I forgot to tell you is when we were learning the generation, we focused on out-of-frame sequences. So sequences that come out of the sequencer, but we know are not functional because, for example, they have a stop codon. So we know that all that ever happened to these sequences, they were just generated. Normal sequences later undergo other selection processes, such as being recognized by pathogens, but also selection processes just to check that they're good functional proteins. So then we look into the first functional repertoire, the naive repertoire, and ask how many receptors do two people have in common. So for example, if I sequence myself and I sequence you, and we can ask how many exact receptors do we share. And so we can look at the data, that's the x-axis, but we can also calculate our random expectation, which is basically the probability that this receptor was generated, which we've now characterized, and now that it was later selected to be functional, which we've also characterized in a similar way, and I didn't tell you about it. So it's just the square of those two probabilities. And we see that the random expectations agree very well with what we see in data. This is for two people. This is the same thing for over about 1,000 people. So we basically ask how many receptors do 2 people share, 10 people share, 100 people share, and so on. And you can see that our model very well predicts what we see in the data. So basically, how many receptors we share by chance share between each other is completely predicted by chance. And you can ask then, well, if we do have this huge diversity to generate receptors, have we generated as humanity every possible receptor there is to be generated? And so this is what we've currently sampled. This is just extrapolation. Of course, we're not complete vampires. We don't suck all the blood out of these people, or if our collaborators don't. This is the model prediction if we extend to the whole repertoire. And we can see, if you look at 10 billion people, this is how many different new T cells each person has. So at 10 billion people, we're nowhere near saturation. Basically, each person will share some receptors with somebody else or other people, and will have some receptors that are unique to this person. And it's thanks to these receptors that are unique to yourself that we will be able to identify you out of the whole worldwide population. And of course, the ones you share with others are the ones that are likely to be made. And the ones that are unique to you are the ones that have the probability of the order of 10 to the minus 15, 10 to the minus 20. So you see how there's this balance in how the repertoire functions. Okay, so while we've been talking about the immune system, there's the other part of the immune system story which guides and leads to the evolution of the immune system, which are pathogens. And pathogens are never idle, they evolve. This is the evolution of the flu taken from a few years back. And you can see that it travels a lot. And in fact, the flu travels by plane. As you now know, so does COVID and so does everything else. So the flu evolution map basically follows the major airline routes. So there is a big question generally, and sort of also for the WHO, how can you predict future flu strains? How can you predict future COVID strains now? Is the predictability in this data? And this is linked to a bigger question in living systems of generally prediction in living systems. So when Serena hits this ball, what she's doing is she's not following the ball, she's predicting the future position of the ball. She's integrating the equations of motion and doing a predictive calculation unknowingly. Another example of prediction in biology are male moths that are born without a digestive system and have of the order of a few days to find a female and reproduce. And they do this in turbulent environments, and there's a question of how do they manage. So one idea that was put forward is that they build a probabilistic map of sources that allows them to predict the position by combining exploration, that means gathering data, with exploitation and a maximum likelihood search. A third example of prediction is the already mentioned flu strain evolution, where basically every year scientists get together and try and predict what will be the dominant flu strain for next year. So they make a decision in February for the flu vaccines we'll get about now, because it takes time to industrially produce them. And the question is, can they actually predict the future strain? This is data that shows the prediction in dashed lines and the later made observation in solid lines. So you can see that by combining stochastic equations of motion with interactions with the environment and existent data from the whole world, they can do that. But we asked a more sort of narrow, well, in a way, a more ambitious question, but also a more small-scale question is, can we think about our own immune systems as systems that are trying to also predict the future strains of viruses and be prepared for when they come? Basically, can they adjust themselves in a way to be prepared for what's coming? Of course, the problem is slightly different than here, because as scientists, we have access to data from the whole wild world. Our immune system only has access to what it has seen itself. And why do we think our immune system has a chance to predict anything? Well, it's because most of pathogen evolution, without talking about systems that come from different hosts, is correlated in time. Evolution is mostly a continuous process in time. So the question is, can the immune system exploit correlations in the pathogen environment to update the frequencies of its different receptors? So for example, add more of these green ones to be prepared for future infection strains. So one thing you need for prediction is to have correlations. If the system has no correlations, you have no chance of predicting. The other thing you need to predict is to actually have memory. You need to be able to remember. If you don't remember anything, then you have no chance of predicting. And so luckily, the immune system has memory. it has a special memory repertoire. So after this story that I already told you about of cells proliferating to have a bigger army to quench the pathogen, some part, the number of cells goes down, that was active, but some part of them is set aside and given a special flag, like you've been to war, and basically set aside in a separate compartment called the memory repertoire that governs its own population size and so on. So the first question we can ask, is this useful? Is it useful to have memory? Because if you have memory, then you sort of want to predict. And second of all, if you do have memory, how should you update it in an optimal way? Meaning, should you update it proportionally? Every time you see a pathogen, you keep the same number of receptors on the side, or should you update it in a modulated way, meaning that the first time you see it, you update it in some way, and the second time you see it, you update it less or more. So basically, you count the number of times you've seen it. Okay, so how do we deal with this? Well, we developed a framework for trying to look at this prediction problem, where we have some pathogen frequencies. We have a distribution of pathogen frequencies, which we'll call QFT, that evolves to some stochastic dynamics that we'll assume we know. We also, on the other hand, have the receptor distribution. So these are the receptors that are trying to recognize the pathogens, and so which also change in time. And the name of the game is to try to minimize the expected cost of an infection. However, there's one problem with this. We don't actually know what the environment is. As I said, we don't know generally what the pathogen environment is. So we're going to assume that the immune system has some belief about the state of the environment. By belief, I mean this in the formal sense of a meta distribution, of a distribution of distributions of the environment. So we're going to say that the immune system has its own representation of the environment, of the current state of an environment, which it dynamically forecasts. So it propagates according to the same dynamics that the immune system evolves according to, which we assume it's learned on evolutionary timescales. And every once in a while, when there's an encounter, when we actually get infected by a pathogen, so our immune system encounters the pathogen, this distribution for that pathogen collapses to the real state, and then it gets propagated further. So basically, we're saying that what the immune system does is it propagates the belief in time, which is a combination of its prior belief about the immune system and the encounters that verify this belief. And it can do that thanks to the existence of the memory repertoire, where it keeps track of what it's seen. So I also have to tell you what the cost is. And for that, I need to tell you a little bit more about the immune system. So the way the immune system functions is there's a very important concept of cross-reactivity, meaning that one receptor can recognize more than one pathogen, a set of pathogens, and one pathogen can be recognized by different receptors. So for example, some pathogen here in the middle can be recognized both by the blue and the red. So we can calculate the probability of an immune response occurring after an encounter with a given antigen or pathogen by just summing over all the receptors that are able to recognize this pathogen, where this are able is basically this cloud. You can think about it as a Gaussian cloud if it helps you. We're going to measure the time in the mean number of encounters, and then we're going to encode the idea that basically the harm caused by a pathogen increases with time. So a pathogen appears, the immune system tries to recognize it and kill it. But if it doesn't, you generally get sicker and sicker. It eats away at your tissues and this incurs a cost to your organism. So we can write this down in this way, that there's a Poisson distribution of recognition events. And then there's some effective cost function for each pathogen that is generally a non-decreasing function of time. It can plateau, but it won't decrease. And different pathogens may have different virulence, may require faster or slower responses. And so this is the cost for one pathogen, and then we have to sum over all pathogens we have. And this is for one value of the distribution of pathogens, and then again we have to integrate over all possible distributions of pathogens. We minimize this and we get the optimal repertoire. So we do this numerically. And so what does the optimal repertoire look like? And does having memory help? So what we're going to look at is the relative cost compared to not having a memory repertoire as a function of the number of encountered pathogens. We can see that the cost always decreases. However, decreases much more quickly for sparse environments. So we assume that both pathogens and immune receptors live in an effective space, an effective recognition space, which you can think about as a two-dimensional, higher dimensional space. And the closer they are to each other, just like on my diagram with these clouds, the more likely they are to recognize each other. And if the pathogens are sparse, then the cost of having memory, the cost decrease, so the benefit of having memory is much larger than if they're not sparse. Okay, so because it helps you to quickly detect the few pathogens. Of course, it also matters how similar the pathogens are. If they're more similar, the cost decreases more quickly. If they're more different, if they evolve quickly, then it becomes harder. But the interesting thing here is that all of these curves collapse onto one curve as a function of this parameter, which basically corresponds to the sampling of non-negligible pathogens. So the two important variables in this problem are sparseness and sampling. And so this is similar to the compressive sensing problem, where these two variables also determine whether we're able to solve the problem or not. Okay, so going with the idea of sparse environments, I see you, is that we see that, well, we can do a sort of estimate basically on how much memory we have. And we can see that even seeing a pathogen 1 is enough to give us a benefit of having memory of twofold. So basically, there is a very large benefit of having memory. And again, what this really means is that we quickly learn, we're able to in childhood, so this is function now of age, this is a cartoon based on data, and this is our prediction. And you see that the idea is that basically you quickly learn the easy sparse pathogens. Your memory increases and your infection susceptibility decreases, and then you plateau in adulthood because you've learned the easy things, and now you're just looking at harder things. And this is verified experimentally. So back to memory management, should you update in a modular way or a proportional way? It turns out that within this theory, subsequent observations count less. You have this negative feedback. And this is also what we see in vaccination. The number of pre-vaccination titers, so basically how much protection you have against the flu before vaccination and how much you have after, depends how big the protection was before. If you had a lot of protection, vaccination doesn't actually change the number of protective molecules you have, whereas if you had little, then you get a big boost. And it turns out that the details of the prior in our calculations, what the immune system assumes has as a belief, don't really matter. You don't have to get the right prior, but just having a prior is helpful. Okay, so to finish, I want to get back to the personalized aspect and actually look at the question of what do immune repertoires look like. So here's a 1D representation of this recognition space. The blue line is the distribution of pathogens. The green line here is the optimal repertoire. You see, we get these peaked solutions that basically, thanks to cross-reactivity, so thanks to these little clouds I talked about, even a pathogen here is covered by the cloud coming from here and coming from here. So you get essentially perfect coverage, although you have these very spiked repertoires. And you get a similar speckle pattern in higher dimensions, such as shown here in 2 dimensions. So it's not the property of the one dimension. So back to our personalized repertoires. If now you and I see the same environment, we of course see slightly different versions of it. We see stochastic versions of this because we sample, but roughly we see the same environment. The question is, should we have the same optimal immune repertoires or should we have different ones? And so the answer was given on the previous slide, essentially, that our immune repertoire should be different, because if this is mine and this is yours, then from afar, they still cover space and they look the same. Although I drew the grid lines here to show you that the receptors are not placed at the same positions. But again, thanks to cross-reactivity, we're able to get perfect coverage. So the similarity between our optimal repertoires as a function of how differently we see the environment very quickly goes down. We have very dissimilar optimal repertoires. However, the antigen distributions we're seeing is the same, and we get perfect coverage, both of us, for each of these repertoires. So we can have very different, phenotypically, we can have very different repertoires and still be protected. And this takes us back to this plot that I showed you, showing you that we can predict the overlap between actual receptors between 2 people. I didn't make a big fuss about the numbers here. The numbers are low. Remember, we're sampling, I didn't tell you exactly, but we're sampling of the order of 100,000, a million cells. Of course, the number of shed cells depends on the sampling depth, but nevertheless, this is a very small fraction. So this is the molecular version of seeing small but predictable overlap. So each one of us finds very different solutions, molecular solutions to similar pathogenic problems. And what I tried to show you is that it makes sense that already at the phenotypic level, we should be able to find different solutions, let alone at the molecular level. So just to summarize, I tried to show you an example of a very complex system, a functional but very complex systems, in which we can nevertheless learn the rules to the extent that we can predict the outcome. We can predict what we expect to see, and I didn't really show you, but we can also predict future states of the repertoire. I then tried to take this into a more theoretical realm of showing you how to think about the immune system as a Bayesian predictor. And what we find is that what an important element in order for the immune system to function well is that it functions in sparse environments. And we find that there's many different optima to this optimization problem, which is something probably relevant also for the discussions today. But in short, even if you have a complex system, a complex stochastic system, you can still make stochastic predictions. And I thank you for your attention.
So we've been hearing a lot about AI over the last 10 years or so, and it's due to the emergence of a new set of techniques that we call deep learning, and it's a revival of some techniques that originally popped up in the 1980s and 1990s and sort of fell out of favor in the mid-90s and then reappeared fairly recently because of some smashing success. And it's all due to a kind of set of techniques that we call supervised learning. So supervised learning is you build a learning system, and this learning system is a program running on a computer, but really it's a parameterized function where the parameters are adjustable. They are kind of represented, symbolized here by the knobs on this machine. And the way you train this machine, the system is that, for example, to recognize images of cars from images of airplanes, is that you show an example of a car, and if the system says car, you do nothing. if the system says something else, like airplane or table or chair, you adjust the parameters a little bit so that the output gets closer to the one you want. So you have to tell the system what the output is, what you want as an output. And then the system sort of adapts progressively. As you show more and more images, the parameters adjust a little bit every time and eventually converge to a configuration where every image of car is correctly classified as car and every image of airplane correctly classified as airplane. And not just the images that you train the system on, but also the images that that the system has never seen, but are similar to others. And this works really well for things like speech recognition, image recognition, generating captions for photos, figuring out the topic of a text, etc. So this is supervised learning. And mathematically, without going into horrible details, this corresponds to finding the minimum of a function. So you can imagine that this this parameterized system I was showing earlier is some sort of mathematical function, g of x, which is an input, and w, which is a set of parameters, a long list of numbers that indicate the position of each knob. And the name of the game here is to figure out a value for all the knobs. So basically values for all the elements of the vector w in such a way that the distance between the output you want and the output that the machine produces is as small as possible on average for all the training samples that you show the system. And for that, we use something called stochastic gradient descent. So what that means is you need to figure out in which direction and by how much to change each of the terms in W, each of the knobs, so that the error goes down essentially at every time step. There's a lot of theory to do around this, a lot of which reuses techniques from statistical physics. And it's a very interesting process. So then the next question is, what do you put in that function? And we call this neural networks because it's inspired by the structure of the brain. So basically, the function I just talked about is a succession of what amounts to weighted sums. So you can think of the system as being a network of very simple elements, and each of those elements, we call them neurons, but it's a bit of an abuse of language. Each of those elements computes a linear combination of its inputs. So it basically uses weights that connect an element to another, and it computes a weighted sum of its input using those weights. And those weights are the knobs that we're going to adjust. So the input-output function of the system is determined by the setting of all the weights, which can be positive or negative. After each neuron has computed a weighted sum, that weighted sum is passed through a nonlinear function, a very simple nonlinear function. And then modern neural nets stack multiple layers of those operations: weighted sum, nonlinear function, weighted sum, nonlinear function, sometimes up to 50 layers or 100 layers, with many neurons at every layer. And then there is forget about the math here. This is not, for those who understand what this is chain rule for multidimensional functions, but you don't need to understand that. Basically, there is a process called backpropagation by which we can, which is based on very ancient mathematics chain rule, which consists in backpropagating signals from the output to the input, so as to compute for every knob, every weight in the system, in which direction to change it so as to make the error on the output go down, and by how much as well. And so that sort of came to become popular in the mid-'80s and really created a lot of surge of interest in neural nets back in the mid-'80s. And then those things turned out to be a little difficult to handle. They required a lot of data to train them. And so they fell out of favor in the mid-'90s. And then they reappeared recently because of various progress in computation and various things. One particular way to organize those neurons is to get even more inspiration from the brain and perhaps organize those neurons in a way that is inspired by the way neurons are connected to each other in the visual cortex in the brain, so that perhaps we can build a system that would be very good at recognizing images. And the classic work in neuroscience by Hubel and Wiesel in the 60s, Nobel Prize winning work, established the type of connections that occurs in the primary visual cortex area in the back of the brain in mammals. And people have been trying to build computer models of that to get computers to recognize images a little bit like animals and humans. The gentleman in the center here is Kunihiko Fukushima, who proposed an architecture called the neocognitron in the early 80s. And then in the late 80s, I proposed, inspired by this, I proposed an architecture called the convolutional neural network, which again is very much inspired by the architecture of the visual cortex, and essentially has groups of neurons that are connected to small areas in the visual field, and then the layers are stacked with sort of several operations called convolutions and pooling. I'm not going to go into the details of that. But this worked really well in the late 80s, early 90s for doing things like recognizing characters, reading the amount on checks, reading zip codes and things like this. And it was deployed actually commercially in the mid 90s. But at the time, we didn't have large data sets of images, and we didn't have computers that were powerful enough to train bigger neural nets than those small convolutional nets. But that changed around 2010, 2012, where large data sets became available to train those systems, perhaps with 1 million samples, 1 million training samples of images of various kinds. And then also GPUs, which are originally designed for graphic rendering, turned out to be very efficient to run those neural nets. And so our friends from the University of Toronto implemented a convolutional net, a very large convolutional net, with several tens of millions of parameters, run it on GPU and trained it on a data set called ImageNet. And they beat all the classical methods by a very large margin. And that really created a revolution where a lot of people in computer vision basically stopped what they were doing and switched to using those systems. So that's pretty much why we hear about AI and deep learning today, because of the progress, those breakthroughs that occurred about 10 years ago. So AI can do pretty amazing things today. There's been a lot of investment in various research groups and companies and startups in applying this to various things like driving assistance, autonomous driving. So every car that will be sold in Europe in the next few years, there's a new regulation that says they need to have basically a system that looks out the window and automatically brakes if an obstacle comes in front of the car, another car, a pedestrian, a bicycle, something like that. Those are called automated emergency braking system. And every car in Europe will have one of those pretty soon. They all use convolutional nets, and they reduce collisions by 40%, so save a lot of lives. There's a lot of applications in online safety and security, which I'll come back on, and then in environmental monitoring, in medicine, medical imaging particularly. In fact, perhaps the biggest application of AI today is for content moderation and ranking online. Both Meta, the former Facebook and Instagram, etc., Google, YouTube, Amazon, they are built around deep learning today. So if you take deep learning out of those companies, they literally crumble. All their services are built around that. And what deep learning does for those companies is that it helps their user deal with the information deluge. It kind of selects pieces of information that are most likely to interest them. It's not perfect. It can answer a question. It can do information retrieval for search, etc. And that requires machine to understand content. It can do translation. So translation works really well these days. And we have systems now that can translate hundreds of languages from any direction. And this is really useful for a lot of people today who can't use technology because for example, they are illiterate. There's 800 million people in the world today who can't read. And so if you have systems that can speak, that can listen, then it's useful. Same for vision. 300 million people in the world are visually impaired. And if you have systems that can explain the content of an image, for example, it's very useful. And then filtering objectionable content, there's been a lot of progress over the last a few years, maybe 3-4 years, in natural language understanding. And that has caused an enormous progress in the ability of online services, for example, to take down hate speech or incitements to violence or bullying and harassment. So, for example, 4 or 5 years ago, only about 40% of hate speech, hateful speech on Facebook was taken down automatically by AI systems. And now it's close to 96%. And that's entirely due to recent progress in AI. It's pretty clear. This is a video which I hope will play well on your side, put together by some of my colleagues at Meta, which shows the capability of modern computer vision systems. So we have computer vision systems now where they can detect any object in an image and put a box around them with a name, estimate the pose of a person, and sort of infer their 3D position just from a single image. And we have systems now that are called semantic segmentation or instance segmentation systems that can basically produce a mask for every object in the image and together with a name, a category. And so this is enormous progress that people didn't think would happen so fast, but because of deep learning, basically that became possible. And the progress has been astonishing. The same type of techniques can be used for doing things like improving the quality of medical images like MRIs. So this is a project, a collaboration between Meta and NYU radiology department that sort of can produce a high quality image of an MRI from a small amount of data. So instead of having to lie down in an MRI machine for 40 minutes, you can only stay for 10 minutes and basically the system will produce an image of the same level of quality as if you had stayed for 40 minutes. And that has gathered a lot of interest in the radiology field in recent years. Deep learning is one of the hottest topics there right now. A lot of applications now of deep learning and AI more generally in biomedical sciences and neuroscience, particularly. So what's interesting there is that those convolutional nets I was talking about are actually used as a model of how the visual cortex operates. And people sort of have ways to explain how the brain recognizes images through the help of those models. Of course, in genomics and certainly in biology and biochemistry. So I'm sure a lot of you have heard of AlphaFold. There's 2 or 3 efforts in the world that with kind of similar performance to compute the configurations of proteins. And this is a big advance that may bring a lot of progress in things like drug discovery, designing new proteins, and things of that type. But it's still work in progress very much. And then also applications in physics, particularly in high energy physics for filtering trajectories of particles, astrophysics in various ways, and cosmology in particular, and chemistry and material science. So what deep learning systems are used for there is to basically produce approximate solutions of equations that perhaps we can solve at a small scale with supercomputers, but we can't solve at the scale of the entire universe, for example. So, some theories of the early instance of the universe to be validated require to basically solve very complex partial differential equations at the scale of the entire universe, and that's not really practical, but you can train a neural net to approximate the solution more efficiently and then have a simulation at the scale of the universe. And there's sort of, a lot of applications of deep learning these days for essentially solving partial differential equations approximately. And an interesting project that was started by my colleagues at Meta is it's called Open Catalyst. And it's an open competition with a data set. And the purpose of this is to figure out if we can use machine learning techniques to design new compounds, new catalysts, or new materials that would facilitate the separation of hydrogen from oxygen. Because if we could do that efficiently at scale, we could store energy, and that means we could cover a small desert with solar panels, produce hydrogen, and then ship the hydrogen wherever it's needed, in the form of methane, for example. And so we currently have techniques, of course, to separate hydrogen from oxygen, but they are either scalable or efficient, but not both. And so perhaps this would help solve the problem. It's an open competition; anyone can participate, and the data is available publicly. So very interesting project and challenging. But in the future, we'll need AI systems that, basically have human level intelligence, because we'd like AI systems to basically help us in our daily lives in the form of intelligent virtual assistants. We'd like domestic robots, we'd like completely autonomous cars, we'd like, many things like that. So how do we do this? And we need machines that have common sense for that. And machine learning today really isn't that great compared to animals and humans. Any human teenager can learn to drive a car in about 20 hours of training, and we still don't have self-driving cars. So obviously we're missing something big in terms of how machines can learn. And what's useful is to look at how babies learn how the world works, basically, in the first few months of life. And they learn an enormous amount of background knowledge about the world in just the first few months of life. They learn, for example, around the age of 9 months, they learn about gravity and intuitive physics. So they learn that an object that is not supported will fall. But before the age of 9 months, they really don't have a concept of that. So basically, just by observing the world go by observation, babies learn a model of the world that allows them to know what's possible and what's not possible, and predict what's going to happen. And what we need to do is get new learning procedures that basically would allow machines to do similar things. So learn like humans and animals. And perhaps this type of learning is what allows humans and many animals to have what we call common sense, the accumulation of knowledge about how the world works, essentially. And so in machine learning, we have this problem similar to physicists, which is that we know how to do two types of learning, reinforcement learning and supervised learning. And then there is this other type of learning that we seem to observe in humans and animals, which I call self-supervised learning. In my opinion, that constitutes the bulk of learning in humans and animals. And so, but it's a bit of a dark matter. So it's kind of like physicists. Physicists they know that dark matter represents most of the mass in the universe, but they can't tell us what it is. And it's a bit the same thing in machine learning. The dark matter of the cake here is what we don't know how to do yet, or we know a little bit, but how to do that is, and I'm going to talk about this very quickly. So first of all, how would we build an autonomous intelligent system? So a system that can, set its own goals and, imagine an outcome of a sequence of actions. And for that, the system will need what's called a world model. So something that, given an estimate of the current state of the world and an action that the agent will take, can it predict the next state of the world? And with such a world model, an intelligent system would be able to imagine the consequences of a sequence of action, and would allow it to plan ahead, to basically plan complex sequences of actions. And the big question is how do we train this world model? This world model basically needs to be trained by observation, also a little bit by play and things like that. And let me go quickly because we don't have much time. And that's the problem that is formulated as self-supervised learning. So self-supervised learning is a type of learning where you only have an input, you don't have an output, basically. So let's say, for the sake of the argument, the input is a piece of video, and you show the system the initial piece of the video, and then you reveal the end of the video. And in the meantime, you ask the system to predict the rest of the video. And by training itself to predict what's going to happen in a video, the system presumably can learn how the world works, that the world is three-dimensional, that there are objects in the world that move independently. Some objects are animate, some are inanimate. So trajectories of objects can be predicted and to some extent, right? So basically getting machines to learn physics, you can think of it this way, but not just physics, also behavior of other agents. So that's the idea of self-supervised learning. You only have an input, you mask a piece of it, and you ask the system to predict the part that is masked from the part that is not masked. And for this, we need to change, this is again a connection with statistical physics, we need to change the way that we represent the predictions that those systems make. So a traditional technique in machine learning is that when you make a prediction, and that prediction can have multiple outcomes, what you're predicting are the parameters of a probability distribution. So, and in the world, of course, you're looking at me right now. I could be moving my hands in a particular way. I could be moving my head to the left or to the right, but you can't tell in advance really precisely what I'm going to do. And so if you try to make a prediction of just this video of my face giving a talk, and I ask you to make a single prediction, that single prediction is very likely to be wrong. And so what you need to predict is not just a single prediction, but a collection of prediction, maybe a distribution if we think in terms of probability. But in fact, that turns out to be intractable. And this is a problem that physicists have encountered at least since World War II, but perhaps earlier than that. And what the technique I'm proposing here is called energy-based models. And the idea is to capture the dependency between two sets of variables, X and Y, let's say the initial segment of a video and the continuation of it, through an energy function. So that's very much inspired by physics and statistical physics in particular. And this energy function will take low values when the pair of X and Y are compatible, and then higher values when they are not. So low value when Y is a continuation for the video clip X, but then higher values when Y is not a good continuation for the video clip X. So it's kind of symbolized on the little diagram here on the right, where the black dots symbolize data points, so observations of video clips, for example. And for any value of X, there can be multiple values of Y that are compatible. And then the energy function is represented by those sort of level sets here, where the energy is low around the data points and then higher outside. And we can turn those into probabilistic models, but we run into a problem that physicists, statistical physicists in particular, are very familiar with, which is that the normalization constant that allows us to turn an energy function into a probability distribution is intractable. And for those of you who know what this is, I don't need to explain. For those of you who don't know what it is, I don't have time to explain. But basically, that makes the probabilistic modeling essentially a computationally intractable problem. And so what I'm advocating for here is to manipulate the energy function directly and ignore the fact that it could be the logarithm of some distribution. And the main problem that we have to face here when we train those systems is to prevent them from collapsing, which basically corresponds to producing an energy function that is flat. And so I don't have time to go into the details of how that, why that occurs, but the way to prevent this is basically two ways. One is to use what's called contrastive methods, which is to generate points outside the region of training data, those green points that you see flashing here on the screen, and then you push their energy up while you push the energy of the data points down. So that shapes the energy surface so that it takes low values on the data points and then higher values outside. Those are contrastive methods. And I don't think they're very popular at the moment for a lot of applications, but I think they're doomed in some way. And then regularized methods, which basically consist in trying to minimize the volume of space that can take low energy. I need to ask you how much time I have left. How much time do I have now? Okay, I can't hear you, but. All right, I'm going to keep going. You're going to have to stop me at some point.
Can you make it in 5 minutes now?
Yes, yes.
Thank you very much.
So a particularly successful application of self-supervised learning is what's called denoising autoencoder or masked autoencoder. And this is what has caused the revolution I was talking about earlier in natural language understanding. The idea there is that you take a piece of text, typically 1,000 words or so, and you remove 15 to 20% of the words, you replace them by a blank marker, essentially. And you feed this corrupted text to a large neural network called a transformer. You train it to recover the missing words and to basically predict what the words that are missing. In the process of doing so, the neural net basically learns to represent the meaning of the text so that it can do this task of filling in the blanks. And this is, very much an example of this problem of filling in the blanks that I specified self-supervised learning was doing. Now, you can use the internal representation of the text that is learned by the system as input to a downstream task, such as translation, hate speech detection, topic classification, etc. And this works amazingly well. I'm not going to bore you with details, but there's very large models that you might have heard of, GPT-3, OPT-175B. So those are very large world models that are pre-trained in a self-supervised manner, and then they can be used for subsequent applications with very little training for things like various text understanding tasks, dialogue systems, hate speech detection, etc. Those are very large systems that require a lot of computations to be able to train. This is one of those systems that is being asked to produce a virtual dialogue between the Statue of Liberty and a person. So you initialize the system by typing text. This is the text in boldface at the beginning. And then you let the system generate the rest of the text. And it will generate a different text every time, depending on a sort of a temperature parameter, another connection with statistical physics. And the system will generate dialogues that in the context makes more or less sense. So those systems are not really that intelligent. They have sort of a superficial understanding of reality, but they're only trained with text. So they don't understand that, they don't know that there is an underlying physical reality under the text, and therefore their understanding of the world is very shallow. You can use those things to produce translation systems as well. So this is a project that was done by my colleagues at Meta recently. It's called NLLB, No Language Left Behind, and it can translate from 200 languages to those 200 languages in any direction. It doesn't have to be trained with gigantic amounts of data, and it works really well. These are the 200 languages that it can translate. Most of those languages, I have no idea where they are, where in the world they are spoken. Even worse for Sardinian. That's one of those 200 languages, a dialect of Italian, I suppose. So let me skip ahead because we only have a few minutes left. And I'd like to, what I think is that future versions of self-supervised learning will allow us to basically take the next step in AI and perhaps build AI systems that, on the model of the architecture that I showed earlier, that will be able to model the world, basically learn how the world works, but mostly by observation. And they're based on what I call this JEPA architecture, joint embedding predictive architectures, which I'm not going to explain. But it's an architecture that can handle uncertainty in the prediction in a very good way, and which also can eliminate complex, irrelevant, noisy information in the input that cannot be predicted. Which is something as scientists that we have to do all the time. And we have a few techniques based on this concept that actually work. One of them is called VICRAG, and I'm not going to explain what it does, but basically it consists in training the system to represent the world with a representation that has as much information as possible, okay? So maximize the information content that the representation contains about an observation of the state of the world, but at the same time contain information that can be predicted. So for example, if you're a physicist, you want to predict the trajectory of planets, you can predict the trajectory of planets by just knowing their mass position and velocity. And you don't need to know their composition. You don't need to know their size. You don't need to know their shape. That's irrelevant. Okay, so this type of architecture I'm talking about here might be able to do this. Basically, focus on important information that can be useful for prediction, and then ignore all the irrelevant details. Of course, I can't explain to you how it works. Ultimately, we can put this in a architecture where those can be used to predict what's going to happen as a consequence of actions that an agent may take. And that would allow ultimately this agent to do planning in a hierarchical manner. And so we're back to this architecture I was telling you about earlier. And what I'm presenting here is sort of a vision of perhaps where I think AI research should go over the next 10 years. And this will require a lot of help from physicists, because the sort of theoretical underpinning of those energy-based models is not completely worked out. And I think much of the mathematics under this is has already been worked on by people like Giorgio. Certainly, there was a lot of contacts between those two communities of neural nets and statistical physics back in the 1980s and 90s, and it's reviving now. There's even more contact now between those two communities. So, so steps towards AI, I think we need to develop self-supervised learning methods that are general. We need to design those, perhaps those architectures that can handle uncertainty in the prediction, and get machines to learn how the world works by observation, like humans and many animals, which will allow them to plan and reason. Thank you very much.